Namaste students. Today I am going to discuss about the topic different classes of antibodies. In the previous video we discussed about the structure of antibodies. In the structure of antibodies there are two types of antibodies, two parts of antibodies, light chain and heavy chain. The light chain have two parts and the heavy chain have five different chains. On the basis of presence of these chains, the antibodies are classified into five types. Okay, there are five classes of antibodies on the basis of the presence of these chains, and the name of these chains: gamma, mu, alpha chain, delta chain, and epsilon chains. Okay, so on the basis of presence of these chains, the antibody can be called as the IgG, IgM, IgA, IgD, and IgE. So first one is IgG. IgG is the one of the most important immunoglobulin. IgG make approximately 80% of the serum antibodies in the blood. 80% of the serum antibody contains IgG. Okay, and the half-life of IgG is approximately 7 to 23 days. The IgG has a basic structure that we discussed in the previous video, and IgG has a basic structure which is monomer structure. Okay, and this is the only class of antibody that cross the placenta. Okay, that cross the placenta, and the newborn baby will be protected by the IgG antibody. So that is the very important thing. So now function of the IgG, what are the functions? It is a immunoglobulin which will provide the immunity to the newborn baby. It is used for the neutralization of the toxins. Ig3 binds to the Fc receptor by the phagocytosis. So before completion of the IgG, I must say that IgG are also of four types: IgG1, IgG2, IgG3, IgG4. On the basis of the requirement, different IgG will be present: IgG1, IgG2, IgG3, IgG4. Next is IgM. IgM is the also important after IgG, and as we know that IgG is found as the one of the most highest in the quantity in the serum. IgM is approximately 30% of the serum antibodies, and they has a half-life of 5 days, and most of IgM are pentamer in structure. We will discuss what is the pentamer structure in the next slide. They are pentamer in structure and has 10 approximately 10. One monomer will have one epitope side, epitope binding side, and they in the IgM there are pentamers, so approximately 10 epitopes will be bound to single IgM antibody. The chain of the IgM is bound by the J chain. Okay, J-like structure. These monomer, the monomer of the IgM will bind each other by the J chain. Okay, it is the first immunoglobulin class which is produced in a primary response of the antigen. So when an antibody will come to the body, then the first an immunoglobulin that will be observed, that will give the response, is the IgM immunoglobulin. What are the function of IgM? It is used for the activation of the classical pathway It is used in the defense against multivalent antigens because it is pentamer in structure. So multivalent antigens will be identified using the IgM antibody. It also used for the opsonization reactions. Opsonance is a technique by which an antigen is coated by the antibodies, and then after coating to the to coating of the antigen, this antigen will be presented to the macrophages and antigen presenting cell for the killing. The next antibody is IgA. IgA make up approximately 6% of the total serum immunoglobulins. So after IgG, IgM and IgA is the third most highly available immunoglobulin, and they have half-life of approximately 5 days, and IgA has a dimeric structure. IgG was monomeric, IgM is a pentameric, and IgA is a dimeric structure. And as we know that two monomer will capture 4 epitopes binding site. Okay, uh, IgA is found in the secretions. That is very important point. If someone asks that which antibody is found in the secretions, the answer will be IgA. Okay, the secretions like saliva, mucus, tears, and milk contain IgA antibody. Okay, only IgA antibody is found in these saliva, mucus, and tears, milk. What are the function of IgA? As we know that IgA is a secretory antibody, so it will provide the protection in the secretory system such as milk, saliva, mucus. So it will provide protection in the secretory system. It is effective against viruses that cause influenza virus, and it is used in the production of infant gut. IgD IgD is another immunoglobulin which contains the delta chain, and they are less in the number. Approximately 0.2% of the serum immunoglobulins have IgD. IgD is a monomer and has two epitopes because one monomer will have two epitope binding site. This class of antibody is found in the surface of the B lymphocytes. What is the function? It is used in the B cell activation. It acts as a receptor for the antigen binding. IgE is the next antibody. IgE has the epsilon chain. Okay, and it is found in very low concentration. IgE, as we know that E for, we can also say that E for allergy also. Okay, it is found in the allergic reactions also. Okay, it contains the epsilon chains and it is used in the allergic reactions, and it contains small percentage of lymphocytes also. What are the function of IgE? IgE is responsible for the immediate hypersensitivity reactions. If the questions arises that which antibody is involved in the hypersensitivity reactions, the answer will be IgE. The IgE will bind to the Fc receptor on the basophil and mast cells and will release some specific cytokines such as histamine receptive mediators, which will cause the allergic reactions, which are responsible for the killing of the allergic reactions and may cause some allergic reactions also, and provide the protection for the allergic and some uh, unnecessary particles which are present in the body. So this is the structure of IgG. As we know that it is a monomeric structure. Okay, this is the monomeric structure and have two portions, two epitope sites. IgE also monomer and has two epitope binding site. IgD is also a monomer and contains two epitope binding site. IgA, as we know that it is dimeric, means there are two antibodies structures which are present in the IgA, and they are bound by a J chain. Here we can find here they are bound by the J chain joining. This is the joining chain here, which will bind this IgA monomeric structures, making it a dimeric structure. Next is the IgM. Here we can find here one, two, three, four, five antibodies, five immunoglobulin monomeric structure mixed together to make a pentameric structure, and this is the joining chain which will join these monomeric structures, making a pentameric structure. So that's all about the structure and function of the antibodies. So our lecture is over here. Thank you very much.